Dear friends, we will continue our lecture of medical diagnostics. So, last time we have seen uh, the procedure of making the thin films. Now we will see the principle, how to make a blood film, how to investigate the blood, how the blood diagnosis is to be made. So, three major steps involved in differential count of WBC. The smear is taken directly from the skin puncture, which gives the true picture of blood morphology. However, EDTA anticoagulant venous blood is satisfactory if smears are made within 1 to 2 hours of collection. Then staining is done with the polychromatic stain that includes multiple colors, that includes methylene blue and eosin in its preparation, or we can say Leishman's or JSMA stain can be used. The polychromatin stain includes multiple colors when applied to the cells. The stains are dissolved in methanol and buffered to pH 7 to 7.2. So methanol acts as a fixative and also as a solvent. So this can be done for the preparation of blood smear to identify or to study the differential count of WBC. So in WBC, white cells and its cytoplasm, which is stained either acidophilic or basophilic or neutrophilic, it depends on the type of dye, and then we can identify the structure, either nucleus or the other contents. Then JSMA stain can give a polychromatic stain, and unlike Leishman's stain, it also gives a violet or blue color stain to the cells and nucleus. How to use it, how to do it? After preparing thin smear of the blood, you allow it to dip under the stain. Then differential cell count can be easily possible for us. Morphologically, we can study the red blood cells also along with white blood cells, but Leishman stain is specially for the WBC or white blood cells, white blood corpuscles. So examination of stained blood smear or Leishman's blood smear. First, examine the stained blood smear under the low power of screening, and this allows you to quickly scan the entire slide. Note the background color and uh, distribution of white cells in perfectly stained smear. So three zones can be identified visually: the thick area of the head of the smear following the body, and finally the thin end of the smear, or we can call it as a tail. The tail end, the red cells lies singly, and neutrophils and monocytes predominate, while in the body the red cells overlap each other to a certain extent, and lymphocytes predominate. If the scanning indicates non-uniform distribution of white cells with larger white cells concentrated toward the tail or edges, then choose the portion of the blood smear usually slightly before the tail end of the smear, where there is only a slight touching of the red cells appears, and place a drop of immersion oil on the slide. Now after oil immersion, we can observe it in a clear way, or we can draw a diagram under the microscope if possible. So identify various types of WBCs from the nucleus and nucleus structure and stain. Then we can find that the granulocytes cells are the appears the granules in the cytoplasm with faint pink stains. Then these includes the neutrophils, eosinophils, and basophils. Then neutrophils pale pink cytoplasm with fine massive colored granule appears and includes bands and segmented forms, normally 3 to 4 lobed nucleus. Then neutrophils are pale pink. It also have 3 to 4 it also have 3 to 4 lobes, and sausage shaped nucleus is appeared. Then nucleus with definite lobes, 3 or 4 lobes, not less or more. Then eosinophils also staining faint pink. They contains large red and red orange granules, whereas basophils are cytoplasmic granules, large and dark and blue black colored. Then lymphocytes are large sized and have clear blue cytoplasm. having on the clear blue cytoplasm on the margin of the nucleus, and smaller lymphocytes dark violet colored. So monocytes are larger in size of all white cells, and wavy margin of cytoplasm is appeared, and nucleus is of kidney shaped. So red cells and platelets are easy to identify based on the size and shape. So red cells are stained pink, whereas platelets mauve pink, or we can say faint pink. So this is the chart given by identification structure or identification hallmarks of the neutrophil, eosinophil, basophil, lymphocyte and monocyte. So this is the diameter of the cells given for identification in micrometer, and fine granules, because granulocytes have granules and agranulocytes have no granules. So cytoplasm is clear. So this cytoplasm shows violet, rosy or purple colored granules, and they are known as neutrophil, eosinophil or basophils respectively. So their nucleus is divided into 3 lobes, whereas in eosinophil bilobed nucleus, and here basophils S-shaped or U-shaped nucleus is appearing. So this is the identification mark of the WBC, differential count. So procedure for differential count includes the observation under the microscope or oil immersion microscope, then examine the thin area as well as thick area as well as the body of the smear, so that you will appear differential count. Then examine the smear under oil immersion, and from the type of nucleus and the granules appear in the cytoplasm, you decide what type of cell it may be, and you report it and calculate it with this formula: number of type of cells upon total number of white blood corpuscles into 100. So you will get the total number of leukocytes present in the body. So these are the normal ranges or normal values in male or female adult. Neutrophils are 40 to 75%, eosinophils are 1 to 6%, basophils are only 1% or less. Lymphocytes are 20 to 45%, and monocytes 2 to 10%. So clinical significance includes differential count is useful to identify changes in the distribution of WBC and may be related to specific type of disorders. It also gives idea regarding the severity of the disease and the degree of response of the body. The neutrophils increases, the percentage of neutrophil is called as neutrophilia, and decrease in neutrophil is observed in the infection such as bacterial infection, viral infection, and other conditions like anemia. And lymphocytosis includes relative lymphocytosis. It is in this condition actual number of lymphocytosis is unchanged, but due to decrease in neutrophils manile, the differential count shows the increase in lymphocytes. So these are the abnormalities which are studied by counting the differential count. Then absolute lymphocytosis, it is found in certain infections such as mumps, cough, measles, influenza, then syphilis, tuberculosis, typhoid, and other chronic infection or infective diseases. And then lymphopenia, eosinophilia, monocytosis, basophilia, these are the causes which are observed due to their abnormal numbers or abnormal percentage observed in the blood. So this may cause the bacterial infection, viral infection, or the diseases like leukemia, MIAS, or we can call it as a typhoid, then influenza, and likewise other diseases which causes the change in the cells, or we can causes change in the percentage of the blood cells. So one of the method of moving the slide for differential count. So this is the start point. We can move up to here. So this is the tail. This is the body. So serpentine counting pattern is generally the middle count. So typical smear is appear in this zone, and we can count the WBC. So this area is giving a clear picture of the cells. And after counting, after observation, you can count the cells which you appear in the under microscope. So you prepare the chart, and then you can calculate it by reporting it. If you found monocyte, you write it M. If you found neutrophil, in this way you can write all the cells: neutrophil, eosinophil, basophil, large lymphocyte, small lymphocyte, and monocytes. So this counting can be possible by making such a squares or this table. So if you will count it as 100, you can get the percentage. So these are the structures which are appear under the microscope, from where you can decide neutrophil, 3 to 4 lobes of nucleus. This is the cytoplasm, and these are the nucleus, multilobed nucleus, and these are the oxophil granules that are appear in the neutrophils. So this granular cytoplasm having dot of pink colored in eosinophils. Nucleus is bilobed, and strongly oxophil granules are found in a eosinophils. And in basophils, the pale cytoplasm, U or S shaped nucleus, bilobed nucleus, and the basophil granules are abundantly found in a basophils. So these are the granulocytes. In agranulocytes, large lymphocytes, small lymphocyte, and monocyte are appearing. So large lymphocyte can be identified from its size of nucleus, which is comparatively about half a number or less to the size of the lymphocyte, whereas nucleus is larger in small lymphocyte, and in monocyte the nucleus is kidney shaped and granules are not there in the cytoplasm, therefore it is called as agranulocyte. And these are the types of leukocytes. So additional information is that the pH of water is of vital importance in differentiating the different types of leukocytes with Leishman stain. It should be between 6.8 and 7.2, and preferably between 7 to 7.2. So care is required to avoid formation of deposits of stain which appear on the film as masses of little black dots. So this should be avoided. Always use the clean glassware. Do not use acid in cleaning. Remove stain deposits with methanol. Always use neutral water for washing. Acid water produces a film that is too red, and alkaline water results in one that is too blue. So neutral water must be freshly prepared, as it becomes acid when exposed to air. So manual leukocyte differential count can be done in this way. So this is pull film and this is push film. So pull film is giving a clear and spread picture, whereas push film is giving a dense picture. So at least 100 leukocytes should be counted and classified. Then ideally 2 into 100 cells should be counted. It is nearly impossible to count more than 100 leukocytes in severe leukopenias. On other hand, in the case of very high leukocyte count, 400 leukocytes should be counted. So this gives the total percentage of the cells. So precision must be discussed again, since leukocytes such as eosinophils and basophils only constitute a small part of the total number of leukocytes. The accuracy of their counts is rather small when only 100 leukocytes are counted. So if the actual percentage of patients' basophils are 5%, for example, the value found by counting 100 leukocytes may be between 2 and 11. So these are only by counting 10,000 cells. Therefore, counting of 100 is average, and this is possible, and this is simple method for the. So this is the chart given by in percentage, then 100, 200 to 10,000. How much cells you are counting? It depends on the. This is called as adjunct table. So it gives percentage of the type of leukocyte to the total number of shaded dark red percentage of the identified leukocytes. So these are the shaded red and shaded. These are the two color bands actually. So these color bands are giving percentage of identified leukocytes, and these are another is the normal. So you can calculate it, or you can put it your values in this table, so that you can get how much is the normal value and how much is the observed value of your blood calculation. So next will be continued in the next lecture.